Hello friends, welcome to my channel Biology with AAGAM Singh. Today we are going to discuss on an important topic, the evidences of the role of DNA as a genetic material. How can you say that DNA is a genetic material and how can you prove that DNA is a genetic material? So to prove this, we have two evidences. First one is by the bacterial transformation principle by Frederick Griffith, and the second one by experiment on the bacteriophage by Hershey and Chase. So we are starting with the first one, that is Griffith experiment. What Griffith had done, he had used rough and smooth stain of bacteria. Rough stain of bacteria is non-virulent because it doesn't contain the mucal or capsule coat. As stain of the bacteria contain the mucor or capsule coat, which shows us virulency, means that it is disease causing. So he had done this experiment in 4 steps. First one, he had injected rough stain of bacteria in a mice. The mice survived. He doesn't die of pneumonia because pneumonia doesn't occur because rough stain is non-virulent. Secondly, he had done inject S stain of bacteria which is virulent in a mice, and he observed that mice die. Mice doesn't survive because pneumonia is opt in it. Thirdly, he had done the heat fixing of the bacteria which is S stain and make it denature and then inject in the mice. The mice survived because the bacteria is denatured. The pneumonia doesn't occur in it. In the 4th case, what he had done, he mixes the heat kill culture of S stain plus the R stain of the bacteria and inject it in the mice. The mice died. He thought, what's the problem in it? I had injected both the stains which are non-virulent. What is the material? How the mice die of pneumonia? How the mice die? After this, he concluded that something is there, some material is there which has transformed the bacteria, the rough stain bacteria in a virulent bacteria, because he had collect from this died mice the live stain of S stain bacteria, while he had not injected the S stain bacteria in a live condition, but S stain bacteria are found in this mice. So he is in a problem that something is there which has changed the property of that mice, which has become virulent. So he doesn't conclude that after it that it's a material. What is the material? But after some time, in 1944, 3 scientists, Harvey, McLeod and McCarthy, the 3 scientists restart this experiment, and they concluded that DNA is that material which changes the property of that particular stain of bacteria, that is R stain is converted into S stain. They done this experiment as they grow a lawn culture of rough stain bacteria and they isolate S stain DNA and then spread it on the rough stain bacterial culture. After the growth, some bacteria which are R stained are converted into S stain. They concluded that this is the material which has something is there which has gone inside. Something what? They had done the DNA now. Then DNA is absorbed by the R stain. The S stain DNA is absorbed by the R stain and become virulent. They prove it by 3 ways. Firstly, they use protease and enzyme which denatures the protein. They use protease to denature the protein. They what had done? Firstly, lawn culture of rough stain bacteria. They put the DNA isolated from S stain bacteria with the protease. The virulency occurs. Means that they sure that protein is not that material which changes the property of that particular R strain. Then they do RNAs. Then they do same with the RNAs. They put the there is a long culture of rough strain bacteria, the DNA of S strain plus the RNase enzyme which denatures the RNA. They do this just like protease. They also get virulency because RNA is also not the genetic material which changes the property of that particular R strain. And finally, they had done the mixing of DNAs. The long culture of rough strain bacteria, they put on the long culture of rough strain bacteria, they put the DNA of S strain plus the DNase enzyme that denatures the DNA. DNase enzyme is that enzyme which denatures the DNA of a particular substance. So after denaturing of DNA, the virulency doesn't occur. They conclude after that DNA is that material that changes the property of particular rough strain and make it virulent. So the conclusion is clear. Now come to the second one, that is evidences on the experiment of bacteriophage by Hershey Chase. The important principle of this experiment is that DNA enters in the cell, not the protein coat. So what they had done in their experiment, that they had taken the two cases in which there is in the first case the radioactive protein coat and in the second case the radioactive DNA. As we all know that in protein sulfur is a part of protein, and in DNA phosphorus is a part of DNA, because DNA is a constituent of sugar, nitrogen bases and phosphorus group. So phosphorus is a part of DNA for the surety that DNA enters in the cell or not. So in both the cases we compare it and study. They do the procedure infection. They infect the bacteriophage and with the cell of E. coli. After infections, the genetic material transferred into E. coli cells. What is the genetic material we are trying to find? And after that, they blend the cell so that the viral particles will detached from the bacterial cell. And after that, they centrifuge it so that the viral particle become as a supernatant and settles down and the bacterial cells goes up. So what we had detected in the first case is that there is a radioactive sulfur 35 detected in the supernatant. Means that protein radioactivity is shown in the supernatant. Means that protein is not that one that enters in the bacterial cell, because no radioactive detected in the cell, as you can see. And in the second case, we had no radioactive detected in the supernatant, but we had get the radioactivity in the cell, that is phosphorus 32. We had cleared from this experiment that DNA is the genetic material that enters in the cell of E. coli, not the protein coat. For identification, we had used this radioactivity so that we can get that which one is the genetic material. After this experiment, we had got that DNA is the genetic material. That's all.